This is TWIS. This Week in Science, episode number 844, recorded on Wednesday, September 29th, 2021. Pump up the science. Pump it up while the science is pumping. Yes. Hey, everyone. I'm Dr. Kiki, and tonight on the show, we will fill your head with footprints, love bites, and heart. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. If you are traveling to planet Earth, a word of warning. Humans! They are a curious case of a creature that does not seem to have found balance with its environment, despite the environment being one of the most hospitable in the galaxy. Meteor strikes are rare, and they have an atmosphere that is not only non-toxic, but also shields them from radiation, allowing them to live on the surface year-round. Gravity levels are a little on the severe side, but only just so. Oh, and they have soil that grows food. Yes, food right out of the ground. Water that self-distributes itself almost everywhere through a network of clouds, and a vast diversity of living creatures that maintain the planet's flora through a myriad of interrelated strategies. The whole system is perfectly self-sustaining while providing abundant resources for the human's existence. What more could you want from a planet? But the humans don't seem to understand how lucky they have it. In fact, it's almost as if they resent the planet that they are on, spending most of their time in self-made cocoons while doing just about everything in their power to make the outside world less habitable. Considerable effort has been made terraforming the surface to make it less self-sustaining, from covering fertile biomes with rock-hard surfaces to killing off pollinators, sometimes intentionally. They have even begun to alter the chemistry of their atmosphere in an apparent effort to cook themselves into their cocoons. So, if you happen to be traveling to planet Earth, beware of the humans. Oh, and while you're there, be sure to catch the next episode of This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I wanna learn everything. I wanna fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I wanna know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Kiki and Blair.
And good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again talking about science that we love, we've discovered, we've dug. We have brought these little treasures to discuss tonight. So excited. Also, we are joined by a wonderful guest who's joined us before, and hopefully will again in the future. But let me tell you what's going on tonight. I have stories about really old life, ancient, growing in the cracks kind of life. I've got some love bites for you all and a little bit of memory news. Justin, what did you bring?
I've got 23,000-year-old teenagers, vampire besties, and just good news, ecology edition.
We always love the good news, as long as it's really good news.
This time it sounds like great news. Yeah.
Yeah. Blair, what is in the animal corner?
Oh, I just have giraffes, rabbits, and fish pee.
And just a little fish pee. Okay. Yeah. It's easy to collect.
Yeah.
Little. And we are joined by our guest, Dr. Bill Schutt, who has written a book, and he's going to talk with us about it. He's going to pump it up, pump the science, pump the natural history of the heart.
Great to be here.
Joining us. Yeah, thank you. So exciting to have you back on the show again. I don't know if you'll be around for Justin's vampire news. I do know from past shows, you do know a few batty things.
I studied vampire bats for about 30 years.
Oh, yeah. Just a little bit.
Yeah.
Yeah.
Yeah. So maybe, Justin, you can,
Mhm.
I don't know, bring in bat news a little earlier.
Tease it out. Okay.
Tease it out. Just work it in. I don't know. Okay. If you have not yet subscribed to This Week in Science, I would love it if you would do so. You can find us all places that good science podcasts are found. You can find us on YouTube, on Facebook. We're on Twitch and we're on Twitter. We're on Instagram. And you can find us some places just looking for This Week in Science. We are TWIS Science on Twitter and Twitch and Instagram. And our website is twis.org. All right. Let's dive into some fun science, starting the show with really old stuff because I'm feeling really old tonight. Okay. But old life rocks. I just want everyone to know that. Old life rocks. Well, I mean, really old rocks that are like 1 billion years older than when the first life was evolved on the planet. They were discovered in Australia, these billion some odd year old rocks that had inclusions of life, traces of ancient, maybe crustaceans, maybe microbes. They weren't really sure. It's a lot of just leftovers from old stuff. But the conundrum was 1.5 billion year old rocks. How the heck was there life in them? And these rocks have been around for, we've known about this issue for about 50 years or so. Like half a century, we've been scratching our heads. Geologists, biologists, paleontologists, like, what is going on with these old rocks? And finally, some researchers at Western Australia and also at UCLA, they have published in the Proceedings of the National Academy of Sciences that these 1.7 billion year old rocks, they cracked a little. And those little teeny tiny microscopic cracks in the quartzite, allowed for the intrusion of life. And so for 50 some odd years, people have been saying, how could there have been life before there was life? And the answer is there wasn't. We just didn't understand how to differentiate. We didn't have the technology to differentiate between the old rocks and the old life. But now we do. And the researchers, this international team, have been able to discover that these old animal fossils, they could have been concrete chewing worms or crustaceans, little tiny crustaceans, but they dug themselves into these rocks early on. The researchers say most likely the traces were made by crustaceans, which invaded southwestern Australia during a short-lived marine transgression associated with the opening of the Southern Ocean. These trace fossils in the wrong rocks have been a mystery. We are glad to have been able to demonstrate geological processes that resolve this conundrum. It's not a mystery. It's geology. We figured it out. It just takes time sometimes.
Those tiny crabs all along.
little teeny tiny crabs. Little teeny tiny crabs in the rocks. Yes.
So
Exactly.
Trauma says, aren't all rocks ridiculously old? What's special about these? So rocks, human lifespan,
Great question.
are all very old. But there's just a few places where the rocks haven't been sort of turned over, covered up, or reintroduced it down to the core of the planet to some degree. There's only a few spots where we have these billion or billions of year old rock formations that are available to us on the surface that we can study. This is one of those few. I think there's another in the Antarctic region, or not, sorry, in the Arctic region, maybe Greenland. But it's very rare to have this billions of year old rock formations on the surface that can allow us to do these peerbacks. Some of the oldest rocks that we've been able to find are below the sea. But for this sort of time frame, it's exceedingly rare.
Exactly. Yeah. They're very old rocks. And it would have been wild for life to actually have been in them before life was alive. Yeah.
Yeah.
Yes. All right, Justin, what do you want to talk about next?
Okay, so this is researchers in Panama attached tiny backpack tracking computers to 50 vampire bats, and some of these were previously held in captivity and studied, and some were completely wild bats. They were all sort of living in a hollowed out tree together, and they tracked their movements. They tracked where they went as they went through their nightly foraging outings. So by day, they all hung out together with a bunch of other bats, and the tracking data showed that the vampire bats were setting out individually to go foraging. These bats weren't going out like you've seen, like the mass exodus of the cave where they all leave together and like, let's all go this way. These ones are just leaving on their own. And the tracking data showed that even though they're leaving separately, they are finding each other in the same hunting areas on the same prey and mingling together with the ones that they're socially the closest with when they're hanging out in the log. The ones that they're socially interacting with, which we've talked about this before too, this sort of, bats have buddies. They sort of have this little buddy system where they'll sort of help out. If there's a sick bat, the neighbor will come and bring some food or something. They help each other out. They become friends in the cave, or in this case, the hollowed out tree. And so part of this story is just they hadn't necessarily done a whole lot of tracking outside of the cave. There's a lot of study of individual bats within like a cave or nesting environment, but not a whole lot of where individuals go once they're foraging. And they were kind of wondering, like, how did this happen? Is this, hey, where are you going tonight? I was thinking about going over that little swamp on the other side. There's some cows over there I spotted last week. I think I'm gonna go visit them tonight. Go feed over there. Hey, you know what? Okay, I've got some laundry to do. I'm gonna vacuum the cave a little bit, but I'll meet you there later, because there seems to be almost a coordinated ability for them to find each other once they're out there. Could be calls, that sort of thing. Except, the distances and the different directions that these bats are going when they're foraging kind of indicates that they have either a routine that they are meeting up to find each other, that they're calling until they find each other, or that they are pre-communicating where they're going to go that evening. Some of the stuff they looked at too is just that relationship that gets built within, persists with the hunting and tracking. Over the course of only 2 weeks, these backpack computers produced data of about 400,000 individual meetings and interactions, which is a pretty decent data set to have gathered in such a short period of time. Another thing they did was they also were able to do some recording in the field, some video and some audio recordings, and they caught some unique vocalizations. Again, a lot of vocalizations have been recorded within the caves when bats are sort of chirping to each other versus when they are there's a buzzing sound, I guess, that is described, that when they're not getting along very well, when they're like, hey, back off, let's work this out. I'm not happy with you. But they had this whole different sort of sounds that they were making while feeding. And so some of these are bats that were grouped together on a single cow, sharing meals, which also tended to be the ones that grouped together in the cave. They spent more time together than bats that they didn't hang out with once they were foraging and hunting. And they were making these unique vocalizations while feeding, meaning that they were also communicating about how well the hunt was going to each other. They were giving each other sort of these updates, whether it was all clear signs that there's nothing dangerous about, or it's like, hey, this is good. What do you think about your steak? Oh, mine's really good. I'm enjoying this blood meal over here just fine. But they had these completely new communications that hadn't been heard before while they were feeding.
So I guess my question here is, Bill, after 30 years of studying vampire bats, was there any kind of inkling that bats would have these kinds of close friendships outside of the roost while feeding?
Yeah, The work that's being done here that Justin described is being done by a friend of mine, Jerry Carter. We both went through Cornell together, and he's basing his work on the work of his mentor, Jerry Wilkinson, who was the first person to show that vampire bats share blood. It's feeding on blood, solely on blood is a tough way to make a living, and yeah. So if you go back to your roost and you don't have blood, and that these animals can starve in 2 days because of that diet. So they have all sorts of adaptations that have evolved that allow them to hunt successfully. And if they don't hunt successfully, to do things like sharing blood. But I had 2 species of these at Cornell for 3 years, and the behavior that we saw every day was just blew us away. And sometimes we knew what it was. They had dominance hierarchy behavior where they would hiss at each other, and they had an aerosol come out of their mouths, which basically meant, I'm the dude, sit back.
They're spitting at each other. They're like, get back.
But they're incredible animals. And I always got the feeling that they were the most intelligent bats that I had ever dealt with before, and it was a whole lot of fun to work on them. Luckily for me, I started working on them in the 1990s. When 99% of everything that was known about vampire bats was known about one species. And the line was sort of, vampire bat is a vampire bat is a vampire bat. And I was coming out of courses that said, if you got 3 animals, 3 different species that live in the same place, then something's got to give. And
Right.
Either one's going to migrate away, it's going to adapt, or one of them is going to go extinct. And so I said, there's got to be differences. And this little guy from the Museum of Natural History took me aside and says, Schutt, you got something now, shut up and go get it. And so I spent the next 30 years showing that there were these real differences between the bats that you saw that Jerry works on are common vampire bats. That's what everybody knew about. The 2 that I worked on were much more rare, and they fed on birds. And that's how they sort of carved out a niche for themselves and didn't compete with the common vampire bats that you just saw. Was by taking to the trees and getting by that way. So their behavior was very different.
Right. So they were specializing on their food source.
Absolutely.
Yeah.
Picturing a bat feeding on a bird seems crazy to me, because even just by weight, like a bat and a bird the same size, the bat's going to weigh so much more.
So we're talking about things like chickens and peafowl that are up in the trees at night in the tropics. So they fly up underneath the branch, and they just bite them on the foot.
Oh my gosh!
And then when
That's
they're done, they fly off.
Yeah, no, I was picturing little sparrows. I'm like, oh my God, they fall right over.
You're picturing the biggest possible bat with the smallest possible bird.
No, they're tiny. Vampire bats are really small.
Yeah.
And, that's one of the things that blood feeders share, whether you're a tick or a bed bug or a leech or a vampire bat, is you're small. And you've got anticoagulants in your saliva, and you're stealthy, and you've got sharp teeth.
The ones that had previously been captive studied, they showed no hunting difference from the wild ones, which means they adapted. And when you adapt that quickly, when you can go from a captive to being back into the wild, it means a couple of things. It means, one, the group has to accept you back. You aren't shunned. You aren't just socially inadept enough to where the group is just like, no, you're not one of us. There's something wrong with you've been captive. You're acting weird. But that they also could learn essentially from the wild group on the hunting strategies and how to get back into the wild and resume that lifestyle.
And that really signifies, like Bill said, the intelligence, the social intelligence that's at work within this species to allow them to work together and to help themselves survive, to help the colony survive, and to make all that work. It's fascinating. Yeah. Thanks for your input on that story, Bill. That was great.
Oh, you're welcome. My pleasure.
All right, Blair.
Yes.
What did you want to bring to us?
Oh my goodness. So we were talking about climate change earlier. I think it was in the disclaimer and stuff. But, when we talk about climate change, we focus on the corals. We focus on, the human element. We focus on migration and pollinators. But we're not talking enough about fish. It's specifically their pee. And that is because fish pee is a really important fertilizer to those coral reefs I mentioned before, but also kelp forests, responsible for a huge output of oxygen on our planet, and also seagrasses and all those areas. Those, the kelp, kind of not exactly a plant, basically a plant. Let's just call it a plant. Seagrass is definitely plants. Coral reefs have photosynthetic zooxanthellae inside of them. So all plant adjacent things, right, require fertilizer, just like plants on land do. But in this case, it's the pee. And so this is a preliminary study, actually looking at the fish pee and its movement in relation to climate change. How does fish pee move? Well, via fish. So as the oceans warm, tropical fish tend to move out of the areas they used to be in, where the waters are a little cooler because it's getting too hot where they are. Sometimes there's clarity impacts. Sometimes there's nutrient impacts. The currents change. All these things impact the temperature and other kind of elements of a watery habitat that a fish is very easily going to move out of if it is not what they have adapted to be used to. And so that's good for their survival, but their waste is excreted out of their cloaca, but also directly through their gills, has beneficial life-sustaining nutrients, including nitrogen and phosphorus, like traditional fertilizers. And so if these fish move away from those areas, the ecosystems can suffer. And so this was, as I mentioned, a preliminary study from the Florida International University. And so Will Weed, a PhD student in Justin Campbell's lab in FIU, in their Institute for the Environment, he collected fish from the ocean and held onto them for about 12 hours, monitored their health, and examined the water that they were in to calculate the rate of excretion so that you could kind of capture potential for an individual fish and what type of nutrients could be contributed from that individual fish. So then based on that, you can look at fish population. And then based on that, you can monitor changes in fish population and try to get an idea of what's going to happen to these aquatic habitats as a result of fish leaving, and therefore their nutrients that they excrete leaving as well. So he's trying to map the larger ecosystem scale, see fish movements, and connect them to their ecosystems via the nutrients they provide. So no major findings yet, except that these nutrients are being excreted in large numbers. And so based on that kind of those statistics, now it's going to be step 2 of that experiment to kind of map that out and map it almost in a 3D sense along with changes in populations and distribution. And so this could be a really helpful tool in predicting and protecting ocean health in relation to climate change. So not any huge findings, but I think a very cool idea for a new tool in response to climate change. Mm-hmm.
Yeah, well, if you're right, so now if you are going to do some attempts to do coral implanting or that sort of thing, you got to know where the fish are gone.
You have to know where the fish went.
Follow the food.
You have to lure them back, and you have to monitor. You have to monitor your transplanted coral reef because they didn't have a reason to be there before. So if they're not hanging out now, you can anticipate a die-off in your coral. So that would be a great reason to monitor that. And if it needs help, you might have to temporarily supplement some of those nutrients until fish established, right?
Yeah, so I was wondering if we could do aquaculture, right?
Mm-hmm.
Take the water from aquaculture that maybe has the pee, the fish urine in it, and supplement a coral reef area. See if that helps.
Or set up a fish farm on top of your new coral reef.
Well, that might be easier than trucking fish urine all over the place.
Yeah, depending where you are. I don't know. It might be kind of far away.
Speaking of fish urine.
Yeah.
Did you guys know that there are parasitic catfish in South America that follow the pee? And that's how they literally will swim up underneath the gill covers of big catfish and attach themselves to the gills and feed on their blood.
Very
So-
good.
So they're
Vampire
using
bats
it
in the
as
sea.
as that's how they're monitoring their prey, basically.
Yeah, they're tracking the
That's
nitrogen.
great.
So if you happen to urinate in the water, then there's
Oh
some,
no.
yeah, they sometimes take a wrong turn.
Oh no!
Bad for the fish, bad for you.
Mm.
Haul out and use the ladies' or men's room is what I'm hearing.
Yes.
Okay, good tip.
You hope so. All righty. You all, if you're listening right now, just want you to know, quick reminder, this is This Week in Science. You're still watching. You're still listening. This is the show I hope you came to enjoy. And I hope you are enjoying it. If you are enjoying it, share it with a friend. If you recall the last appearances of Dr. Schutt, he has joined us on the show to talk about blood-eating animals, cannibalism, all sorts of fun topics. He's an emeritus professor of biology at LIU Post, research associate at the American Museum of Natural History, zoologist, and is here with a new book, Pump, A Natural History of the Heart. Welcome back to the show, Bill. Thank you so much for joining us again.
It's great to be here. This is one of my favorite shows to visit, and to be interviewed by you guys is just a lot of fun.
Oh, well, I hope we can
Thank you.
let you have more fun as we're keeping you up late tonight. So you've gotten bloody, you've gotten cannibalistic, you've done all these things. It seems like a logical path to the heart, but I mean, I need to ask, was the interim kind of a straightforward, oh, this is the next thing to talk about?
Not really. I had to sort of be talked into it. I wanted to do stuff that was a whole lot more vile, and I got talked out of it by my wife, who said that I'm not going to live with you for 2 years if that's what you study. And my, from a sort of financial perspective, my agent and my editor at Algonquin suggested that I look at something a little bit more mainstream. The Cannibal book just did very well, and I was thrilled with it. And I'm still getting interviews about that one. But they wanted me to do something that was that more people might pick up. And so they suggested the heart, and I thought, well, that's got to have been done. And there are hundreds of books written about the heart, and so I did a bit of research. And the type of book that you're familiar with it is. I go through the animal kingdom and I look at these interesting stories, and I'm telling, I'm educating, and I'm trying to be entertaining and inserting humor where appropriate, and then move into humans and myths and history. I'm a big history buff, and so that book just hadn't been written. So I found, I thought this is not going to be weird enough, and I was completely wrong. That there are things here that rival cannibalism as far as like weird behavior of folks thinking that they're doing good medicine back in the day, and then in the animal kingdom itself, there were a lot of neat stories, and the cool thing was that they tied into to cardiac medicine and especially the future of cardiac medicine. So I had a really good time. The learning curve was really steep, and it was a lot of fun to write.
But as a biologist, as a zoologist, I mean, if you're a biologist, of course, at some point you've had basic physiology. You understand the heart, it pumps, right? So, I mean, I went through neuroscience training, and so along the way, I got to learn that once upon a time, we looked at the brain as just a bag of goo that was meant to cool the blood, and that the heart was the center of our emotions and our intellect and all these things. What, as you were discovering all this stuff, what were some of these key moments in the development of our understanding?
Yeah, that was certainly a question that I was interested in. Where did that whole idea of the heart as being the center of the soul and emotions and intellect, where did that come from? So when I was writing this cannibal book, I was interested in where did the cannibal taboo come from? When you say the word cannibal, you have this sort of knee-jerk reaction.
Yeah.
And so the name of that chapter was going to be Blame It on the Greeks. and they changed it. I guess they didn't want me to get stoned by people, Greek people. So Blame It on the Greeks got changed to a different name. But when I started to look up how this idea of cardiocentrism took place, I could have named the chapter "Blame It on the Ancient Egyptians," because it was really them. They thought a great deal of the heart, and as you said, that not so much the brain. When they were mummifying somebody, they would take a hook and shove it up their nose and yank the brain out.
Get rid of all that useless stuff.
Yeah, and they thought that they saw the heart beating. They saw that the heart could react to different situations by speeding up or slowing down. And the sort of natural response to that was the belief that this was the center of everything. And so what happened was that this got passed down to the ancient Greeks, who paid a lot of attention to what they learned from the Egyptians. And so some high rollers like Aristotle and Hippocrates, they were in on this idea of cardiocentrism. And at the same time, now you have, if that's what you're thinking about, and they also got a lot wrong, the Egyptians, and the Greeks picked up on that. They thought that arteries carried air and that veins carried blood that came from the liver, and some of it passed through invisible pores and in the heart to the left side, and then mixed with, pneuma, this sort of spiritual essence that was in the air. And so they got a little bit wrong. Yeah.
Just a little.
Yeah. But I'm not making fun of these ancient cultures, because they did not have the same type of technology, and that they did get a lot right. They weren't able to dissect humans, so they did their work on animals for the most part. So at the same time the Greeks picked this up, then you got artists moving in and picking up on this idea of cardiocentrism, and poets and writers. From there, the Romans picked it up. The problem here, though, was that one of the Romans that studied anatomy, and he wrote 3 million words that were eventually translated, was Galen, and Galen shows up in all 3 of my books, and the problem was not that Galen was a bad surgeon. It was that he got a lot wrong, and his works were, after the fall of the Roman Empire, that his work wasn't initially translated into Latin. So it sat, and in the early medieval times, Syrian Christian translators translated Galen's work, and they put their Christian slant on it. And so when those translations got translated finally into Latin, the church fell in love with it. They thought, well, this is science that we can live with. And this had to do with all sorts of different, these were things like, the 4 humors, and you would bleed someone in order to balance these humors, or, you'd give them emetics. And for 1,500 years, he literally, his teachings caused a stagnation in the field of medicine that just carried on for this unbelievable amount of time. No one did any work. You just referred to Galen. That to me was problematic.
That it would last for 1,500 years. I mean, that's having an impact on a field.
Oh yeah. And it wasn't until the 17th century, at least in the West. In Eastern medicine, they certainly made advances in the Middle East, and in places like that, and in India. But in the West, no. It was bad news, and until folks came along that discovered how the heart circulated blood, that it was really a double pump. You had one side sending the blood to the lungs and back, and the other. And this is William Harvey, of course, and then the other side pumping oxygenated blood out to the body and then back. And from there, the advances happened pretty frequently until we had a much better understanding, but we were still bleeding. Folks were going to barbers and getting bled right through the 19th century. And of course, there was
to barbers.
Yeah, barbers. Barbers did minor surgery. And, if you look at the old time barber's pole, the stripes, the red was blood, the white were bandages. The bulb at the top was where you kept the leeches. So yeah, they did all sorts of fun stuff. So you go get an enema from your barber. It's been years since since I did that at a barber.
Shave and a haircut and a bleeding. Right.
Oh my goodness. Well, their business has been cut into a bit by the advance of medicine.
Yeah. So along the way, we had these beliefs that kind of were perpetuated for a long time through the church, and it helped them along, and then medicine and discovery started making advances when we discovered, like you said, the pumping action of the heart. This is common across all animals that have a heart. Yes?
Yeah, I mean, listen, some of the things that you can call a heart, if you were a cardiologist, these are not card carrying hearts. These are muscular pumps that pump up fluid around the body, whether it's an open circulatory system where the stuff gets dumped into a chamber and the tissues get bathed in it, or it's a closed circulatory system like vertebrates have, fish and amphibians and reptiles and mammals. But the idea is the same: is that you can sort of demonstrate this if you had a water balloon and you wrap your fingers around it, and that your fingers are the muscle fibers, and if you squeeze on that water balloon, you're going to send water bulging out one side. You can think of that's how this pump works in its simplest form. One of the things that I had to emphasize, and I try to do this all the time, is that we have this bad habit of thinking that all of the anything that's not human is either primitive or defective. When I was a kid, it was Neanderthals. They were always dragging their women around by the ear or the hair, and they had a club and they were hunched over. And we know that that's just garbage. And so
projectionism.
Yeah, it's really the same thing with organ systems. We think that we've got the best stuff, and the take home here is that no matter how simple these structures are and how different than ours, they all work perfectly well for the creatures that where they evolved. So we need to sort of get that prejudice out of our system.
Justin likes to remind us that we are everything on Earth equally evolved. We all started
at the same point.
Yeah.
I've only talked to one heart surgeon. And I was at the time surprised to find out that he hadn't studied medicine. That he was a mechanical engineer who had been studying fluid dynamics was his specialty, and that's what he was doing for almost entire education, and kind of fell into heart surgery at the end when he discovered that everything that he knew about mechanical engineering applied to the heart.
Yeah, absolutely.
Yeah.
No doubt. It acts like a machine. So, I talk about the fact that the hummingbird heart probably maxes out. You're not able, we don't think that you could have a heart beat faster than 1,250 times per minute. That's ridiculous.
I can't even move my fingers that fast. Like that's to flutter.
Yeah. So in order to pump blood to the wings that are beating hundreds of times a minute, there's got to be another way, and the way they do that is they've got a heart that's eight times the size, relative speaking, than the blue whale heart that we looked at in this book.
And I love how you start your prologue talking about whales. Mm-hmm. I love that using the largest animals on the planet is how you start the exploration of the heart. I mean, I'll let you explain the perspective that it starts out from. It's not just the heart.
It was sort of tragic because 9 blue whales washed up in Newfoundland in 2014, and nobody really knew a whole lot about them. My friends at the Royal Ontario Museum, they got asked questions because they had whale exhibits, and they were like, "What's the largest heart in the world?" A blue whale. How big is it? Probably the size of a sedan. But nobody really knew because blue whales, when you throw a harpoon in the old days, sunk. So they were the wrong whales as opposed to the right whales, which is the ones that didn't sink.
So this, I did not know that that's where the right whale name came from.
Yeah.
It's the wrong whales and the right whales.
Yep. Well, these are the wrong whales because they could swim at 35 miles per hour, and then when you killed them, they sank, which sort of was a bummer for you if you were a whaler. But they were able to recover because 3 of these didn't sink. They were two of them washed up in these isolated villages on the coast of Newfoundland, and So there was this huge project to recover these hearts, and they brought in construction equipment, and they put 4 people inside the whale to push the heart out of the whale. They'd never seen anything like it. And when they got it outside, when I looked at the pictures of it, it didn't look like a heart. It looked like a giant, a 400-pound soup dumpling is what it reminded me of. And it was so different than say a beef heart or something like that, which has got shape to it. This thing just collapsed. And they think that it has to do with the fact that they dive so deeply, that this is an adaptation for deep diving. The heart doesn't have to beat when you dive down that far. That they can slow their heart rate down to 2-3 beats a minute. So there was so much that they learned about this. So we were always worried about, when we were in lab, that we'd splash some formalin or formaldehyde on ourselves. And here they were worried about falling into a vat of 500 gallons of it.
Yeah, they're trying to fix all those tissues so they don't degrade.
Yeah, so
yeah, that must have smelled lovely.
Yeah.
So I'm remembering I'd use formalin formaldehyde to fix bird brains in graduate school, and we'd leave them overnight. How long did they fix this whale heart with 500 gallons of formaldehyde?
These processes, from the rinsing to the actual preservation, each took months. It was they kept it frozen until they figured they could get people to work on it, and then eventually they sent it over to Germany, to this place. You're all familiar with the bodies exhibit, where they plastinate. So you'll have a guy who's like dribbling a basketball, but he doesn't have skin. Yeah. This was the largest project that these guys had ever undertaken, and so if you look at the finished product, it's an incredible work of art, but it's a whole lot smaller than they thought it was. So they thought they were going to have a big SUV sitting next to it in the exhibition hall, and really they had like a little mini car. So it's sort of golf cart size. Yeah, there are blood vessels there that they don't quite know what they are. And look, the heart is bifurcated at the bottom. There was just so much about it that was really different. But the heart size was the thing that surprised them the most. If this was a hummingbird the same size as that blue whale, the heart would be 8 times larger.
So it's more efficient for the whale's size. And is this something to do with the whale's blood, the whale's physiology? I mean, there's always trade-offs in the form and the function of animals. So it just makes me wonder what aspects of the physiology led to this difference.
Yeah, I think you nailed it, and it probably, as I mentioned, the way that it doesn't sit solidly, if you're looking at it, has to do with diving, and the fact that it's smaller probably has to do with the fact that the metabolic rate of the whale is much lower than, say, a shrew or a hummingbird. So it doesn't have to be as large. It doesn't have to pump as much blood as often. So this is what's evolved over millions of years, and it works perfectly fine.
Did you look at all into like differences in oxygen carrying capacity of blood and how that affects? So if you have blood that can carry more oxygen, can the heart be smaller so that it doesn't have to pump as much blood?
The only instance that I ran into with that, and there are probably a whole bunch that I missed. I looked at ice fish, which live in the Antarctic, and it's absolutely freezing. But their blood is crystal clear, and they don't have hemoglobin. So it's like, how did they survive? Their hearts are really large, and their blood, they have 4 times more blood in their body than a fish a normal fish the same size. Plus so it can carry more oxygen. Plus they have no scales, so they can absorb oxygen through the water, and cold water carries more oxygen. So they have all these adaptations. So there was a mutation that took place, we think, 5 million years ago, where suddenly they didn't have, some of them didn't have hemoglobin. And if they were anyplace else, they probably would have died immediately, because they lived in this really cold area. And they're not really active. They're sort of like sit and wait predators. They don't really have a lot of enemies. So they don't really have an active lifestyle, but they've got all these adaptations now, including what you mentioned, blood that can carry more O2 than a typical fish would have.
But without hemoglobin.
Yeah. Yep.
That's fascinating.
Yeah, it's weird. So their blood is clear, or nearly clear.
That's when we think of blood, it's like it's just going to be red, right? And it's okay. So we've got clear blood. We've talked on the show, Blair, about the blue blood of the
horseshoe crabs.
the horseshoe crab. Yes.
Yeah.
Are there any other colors we need to be aware of?
Blue and red cover it, and it has to do with copper. If you look at the Statue of Liberty and it has that sort of blue tint, that's what happens when copper oxidizes. When you add oxygen to copper, it turns blue. When you add oxygen to iron, which we have in hemoglobin, it turns red. So that's pretty much it.
Was there anything else like this? We've got whales, we've got hummingbirds, we've got different kinds of blood. Was there anything else that was just really surprising to you as a zoologist as you were writing the book?
Well, a lot of it had to do. I mean, I wrote a lot about about medicine, and some of the things that are coming down the pike are just blew me away. And this has to do with folks like Harold Ott at Harvard. There's a real demand for donor organs, whether it's hearts or kidneys or livers, and a lot of people die on waiting lists. Thousands of people die every year waiting for a transplant for reasons that are pretty much commonly known about tissue typing and the fact you've got to ship this thing across continents, et cetera. But he's trying to get around that by taking cadaver hearts. So people were like, "Were they going to rob graves?" I'm like, no. If you have it written out that if you die, that you're instead of your heart going to a medical school, that it's going to be a donor heart. And they take these hearts, and what Dr. Ott is doing is putting them through this rinse, and it's literally a type of detergent that washes away all of the cells that your body would reject if they had taken that donor heart and transplanted it into you. So what you're left with is something that looks white, pale white structure made of mostly of collagen. So it's a connective tissue that you don't have an immune reaction to. And then he's taking stem cells, and this technology exists. Taking cells from the person who'd be the recipient, from their skin, fibroblasts they're called, common skin cells. So they don't have to go into you and do a biopsy or anything like that. Converting them into stem cells, stimulating those stem cells to become cardiac muscle cells, and then seeding this scaffold, this model that's left of connective tissue, and growing a heart that they will then transplant into the recipient. They think about 10 years from now they'll be doing that.
I think if they can get to that point, it's going to be amazing. And the amount of progress that they've made in the last 10 years is astounding. And this technology just keeps advancing in leaps and bounds. They've been able to start getting blood vessels. And this is also research that's part of fake meat research, right? You want a steak that's grown in a petri dish? Well, it's got to have blood vessels in it and fat marbling to make it more real. And all of this is the stuff that needs to go into growing a heart too.
Mm-hmm.
Yeah, all those pieces are going to come together. The idea that at some point, Blair, you want to live forever? We'll just grow you a new heart.
Yeah. Well, that's the next step, right? Is instead of taking a donor heart and bleaching it out, is to just 3D print the collagen.
Yeah. They're doing that in Israel.
Yeah, I can't wait. I can't wait for my factory fresh organs someday.
They're also growing blood vessels because it's really tough to do veins. Arteries have got that muscular layer, so they're easier to knit together if you're doing surgery. But veins are problematic because they're so thin. So I go to this lab at Worcester Polytech Institute in Massachusetts, and I met this guy, Glenn Gaudet. And he comes out, he's got a plate, like a salad. So I said, it's a little early for lunch, but thanks. He goes, oh no, this is what we're using. So he takes a spinach leaf and he shows it to me. He goes, you see the veins? I go, yeah. He brings me over to his lab bench, and he has these spinach leaves hooked up to these drips with the same type of detergent dripping through them. And what it's doing is washing out all of the cellular material and leaving cellulose. And there's your vessel. So now you take human vascular cells and embed them onto that tube, and you tell them to grow around it. So that's what they're doing.
Wow.
Because
How
I'd asked-
cool.
That's amazing.
yeah, I'd asked Ott. I said, "Well, how are you going to? You've got this heart here. How are you going to deal with the fact that you've got coronary vessels that need to supply it?" And he said,
Mhm.
"I'm sending you to a friend of mine." And so the next day, I met this guy at Worcester Polytech, and and he showed me. And that was from a graduate student of his who came up from lunch, and they actually used the spinach that they got in the cafeteria. So, I mean, that's the kind of stuff that, when you asked, was there, or when people ask, was there enough to write about in the heart? When I'm used to writing about cannibalism and blood feeding, yeah, there was plenty, and that was, there was a whole lot more than that.
Was there anything that you left out that you wish you could have put in, or that now that you're like looking at it from now that the book's published, oh, I should have kept that in there?
Yeah, well, I would have, but my editors thought that it was a little on the gory side. So, make a long story short.
Perfect for this show.
Yes.
Long story short, if you look at a horseshoe crab, right? So if you look at our hearts, they are the beat itself is governed not by nerves that come in to stimulate the heart, and every time a nerve impulse comes in, the heart contracts, right? But there's a pacemaker that does that. It sends out a signal from within the heart. So horseshoe crabs are not like that, and many animals, they have hearts with a nerve bundle outside the heart that then sends a signal down to the heart, and the heart contracts. And so I thought, well, this is a really good, I could open up with this really snarky line about, this is why you never see the Aztecs on top of their pyramids holding up a horseshoe crab heart and it's beating, because it wouldn't beat. As soon as you cut it out, it would stop beating. So I had this whole scene written about, this prisoner who got, it was really cinematic. It was like writing my novels again. They marched this guy up. And meanwhile, I'm now explaining how the sympathetic nervous system works, because his heart is beating faster, because all of these stress hormones are getting dumped into circulation. And he gets up there, and they finally, he goes through this entire process, and it ends with the priest holding the heart up and it's beating. But my editors went, no.
Use that when you write your next novel. Not this.
Yeah. So yeah, that's why I was laughing when I heard that, because that was about 1,500 words that got yanked out. But
Nothing new when you're writing.
I think that they thought it's kind of slowed down the, it was kind of like when Kubrick filmed when they did The Shining, they actually filmed the scenes in the maze, which I thought was the best part of Stephen King's book. And then they cut it because they thought it would just stop the movie cold and people would be so freaked out. And I think that was probably what happened here. Not comparing myself to Kubrick or Stephen King, but same idea.
I don't see why you shouldn't. Just it's good. If people were to read your book to get an understanding of the natural history of the heart, how can it help people kind of understand our modern understanding of the heart and things like heart disease and cope with our modern setting?
Oh boy, that's a tough one. There's been a lot written about now what we should do, because we have the wrong diet, and a lot of us are sedentary, and we need to do these things. So I go into that as well. I talked to this guy, Dr. McBride, who was a specialist in post cardiac rehab. And I said, so what works? And he gave me this long list of things that work, statins and meditation, or he said spirituality. And I went, But he also said red wine and dark beer, and I celebrated.
Yes.
But everything in moderation was what he basically, that's what he said. You don't want to go run 50 miles. You want to walk every day, and you don't want to drink 3 gallons of wine. You want to have a glass of wine, and a lot of that having to do with the fact that our hearts have evolved to not deal with. They haven't evolved to deal with the kinds of stuff that we pour into our bodies, and whether it's recreational or dietary, and then the fact that we sit around a lot and don't exercise, that's problematic. So that was kind of interesting. And he went through all of these things about cardiac rehab that I'd never known. 1 in 4 people go to cardiac rehab, and those are the people that survive. A lot of times there's a lot of baggage that comes with a heart attack, and it's things like depression. So I get into that a bit, that whole idea. There was a major study done that said that if you had a heart attack and you were then clinically depressed afterwards, you have a 17% chance of dying within 6 months. And if you had a heart attack and you were not clinically depressed, you had a 3% chance of dying.
Correlation is not causation, but at the same time, those are statistics to take to heart. Oh, yeah, this book is fantastic. I've been reading it, I'm really enjoying it. As a physiologist, I'm loving it. Comparative physiology, it's fantastic. And then also, like you said, covering the modern medicine and the how we think about and what's going on in our bodies and our hearts and how we live today. And it's a natural history of the heart. You've written it, you've done it, and you've definitely done it justice. And thank you for joining us for another.
This is a treat for me, believe me. This was a lot of fun. I really enjoyed hanging out with you guys. I wish you the best of luck.
There'll be another one, I hope.
Oh, yeah, on teeth.
On teeth, fantastic.
On teeth?
Yeah.
On teeth.
Oh, wow.
That's chompers.
What I'm working on now.
And I, in the meantime, will work on as many chewy puns as I can.
It's gonna have all sorts of teeth. Are fun. They've got all sorts of forensic angles on them too.
Fantastic. And this book is now available in all bookstores?
Yes, everywhere that books are sold, in any kind of format that you want.
Great. And your website is billschutt.com. Bill, SCHUTT.com. We will have a link to it on our website and to your Twitter account. Is there any place else that people should go to find you?
On Facebook, Bill Schutt, author. I'm around, and I love talking. If people ask me questions, I'm always willing to try to answer them. I like talking to my readers. But this is not a textbook, so that should be a take-home message.
There are a lot of stories. It's good. Yes.
Thank you very much.
Thank you so much for joining us. Have a wonderful night.
Thank you.
Thank you. Take care. We still have some more twists to come. This is This Week in Science. If you enjoyed the show, please consider heading over to TWIS.org, where you can support us through our Patreon community. Click on the Patreon link and you can choose your amount of support, $10 and more per month. And we will thank you by name at the end of the show. You'll also receive some other goodies by mail. You know that TWIS is listener supported, and we also have our calendars that are available on the website now for order. So if you are at twis.org and you're not really feeling the Patreon thing at the moment, you can click on our calendar purchase button for order. We will be getting those out to people before the holidays, we hope, because the mail system is going to be crazy. But anyway, it is now time. We're going to come on back with This Week in Science to the segment of the show that we know and love as Blair's Animal Corner.
With Blair.
Yes.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels.
What you got, Blair?
I have giraffes.
Woo!
Yeah. So this is a story from Pennsylvania State University looking at giraffe social dynamics. So obviously knowing social dynamics are pretty important. We talk about them on the show all the time. When we know how animals are connected to one another in social networks, that influences things like reproductive success, population ecology, information spread, and sometimes most importantly, how diseases move through a population. All important things to consider, especially when giraffes are endangered. And in this research team, they examined social connectedness and social movements of endangered Maasai giraffes in the Tarangire ecosystem of North Tanzania using data collected over 5 years. They constructed a social network of more than 1,000 free-ranging giraffes during that time. What they found was that male giraffes overall had higher social connectedness than females, which is kind of surprising because, do either of you know the makeup of a typical giraffe herd, or a tower of giraffes, as they are sometimes called?
So first, a tower of giraffes.
Yeah.
Love it.
Yeah.
And second, no idea.
Oh.
No
So,
idea of the makeup.
Much
I mean,
like
unless it's like blue eyeshadow. No.
Oh, yes, makeup. No, much like a lot of hoof stock, there is one male, many females. Males that are born to that group are expected, once they grow up, to skedaddle. And females usually skedaddle until there is a changeover in the male, right? And then they can stay. But ultimately, that is how this works. So you would expect social connectedness in males to not be very high because there's one of them and then a bunch of females. You would expect, conversely, that females would have very high social connectedness because they have to work together within their herd or tower. But this found kind of the opposite. Male giraffes have higher social connectedness than females, and that means that males interact with greater numbers of other individuals than females. So the older males had the shortest social path lengths of all the giraffes, and this might reflect mating strategy of those males. They roam widely across landscapes, searching for females, searching for a group to join. And so young males have the most social ties because they're playing the field and they move the most often between groups. So so that part makes sense. But what's interesting is that these adult female giraffes have fewer but stronger relationships with each other than males and younger females. And so other research from the same group previously found that relationships among among female giraffes allow them to live longer. So the females with stronger social connections actually live longer. Again, correlation, but they found that. So the reason that this kind of maybe perhaps is the case is that this very large study found that as opposed to previous research on giraffes thought that there were adult females. They have about communities of 60 to 90 individuals. A specific region might have a dozen of those 60 to 90 individual groups. They associate more with people within their group of 60 to 90 than with members of other groups. That was the considered kind of understanding of how giraffe social structure works. However, this new study brought light to a new dynamic. The female communities are embedded within larger groups called super communities. So these females are talking just to each other in these smaller groups. But there are groups of around 800 to 900 individuals in these super communities. And so there's lots of transfer of communication and interaction between these little tiny communities within the super community. And that is why you're seeing this increased number for males, is they're hopping from group to group trying to find the right place to kind of get on top. And so they have more opportunity to talk, to socialize with other giraffes while they're trying to find where they fit in. And so they're floating around. There's social butterflies. Meanwhile, the females are kind of more cliquish. This is great because it allows gene flow between groups. So this prevents any potential inbreeding and allows for this kind of gene flow, which is great because, again, giraffes, no matter how you categorize them, 7 subspecies, 4 subspecies, we've had that conversation before. Nobody knows. This might be part of the reason that they're having so much trouble categorizing these guys, is that over the millennia, they have hopped around so much and there's so much fluidity that giraffes could be kind of like a ring species where they have genetic flow all over Africa. Yeah, that's like not it's causing these kind of blended lines across subspecies, which I think was my own kind of interesting that I brought out of all of this. But of course, this is all really important because, again, giraffes are endangered.
Yeah.
And if we understand their social connections, they can have a more successful time in their conservation efforts. So they don't want to translocate animals into new areas if they are part of a super community that they are removing them from. If they understand gene flow within these super communities, that will help them decide where to bring in new genes from that might not be representative. They're trying to beef up the gene pool, all these sorts of things. But anyway, giraffe herds, towers, whatever you want to call them, are a lot more complicated than we thought. And there's a lot more movement amongst those smaller groups than we thought.
Which is good to know.
Mm-hmm, mm-hmm.
I mean, but as it comes out, how often do we say it's more complicated than we thought?
Yes, always.
Almost all the time.
Always. Speaking of things we always say.
Yes.
I have another story that I have a little bit of a rant, but I'm just going to give you the story first. I'm going to try to not inject my own narrative. It's going to happen, but I'm going to try my best.
Okay.
This is a study from University of East Anglia, and it's part of a really important study looking at endangered animals in Europe. So there's a thing called the Shifting Sands Project. They're one of 19 projects across England that make up the National Back from the Brink initiative. And so together, they are aiming to save 20 species from extinction, and by doing that, benefit over 200 more. So it's a really, really great cause. They are trying to take a smart approach to this. They're not just kind of throwing money at the problem. Research is involved, so they can figure out how best to prioritize and save these animals. And so this study in particular that I brought tonight is focused on rabbits. This is specifically looking at wild rabbits in England, and their numbers are declining regionally, nationally, and globally. They have recently been classified as endangered in their native region, which is the Iberian Peninsula. So rabbits, we think about something that's all over the place. And part of the reason for that is that there are pet rabbits. So we're just used to seeing them all the time. The idea that they could be endangered seems crazy. But one of the reasons these rabbits are endangered in England is from viruses being spread from commercially bred rabbits. So people have rabbits as pets, or maybe they have them on a farm or any number of things. And a rabbit will get out. And if you see rabbits in the wild and then a rabbit got out, you're like, it's fine. They're rabbits.
It's rabbit. They're all rabbits. It's fine.
They're not all the same. Not all rabbits are the same. They come from all over the world. A lot of pet rabbits and commercially bred rabbits are not native to whatever area they're being sold in. And so it causes all sorts of problems. Not to mention, if you are a farm and you have rabbits and you're responsible about not letting them get out, they're still, depending on your containment, if you're scrubbing out your boots after you leave your space with them, you can spread disease into the wild land without realizing it. So all that to say, these rabbits are in trouble. And so the reason that this group wanted to look at rabbits is that they claim, there's my hint, they claim that rabbits are keystone species. They hold together an entire ecosystem by grazing and digging.
So by the fact that you said they claim as opposed to they reported or stated or they are, is because I'm assuming you have a little bit of hesitancy accepting this information.
Here's my thing. Here's my thing about keystone species. When I first learned about them, I thought they were so cool. I was like, yes, this makes total sense. Otters, keystone species. Elephants,
beavers,
keystone species. Beavers, keystone species. There's
pandas.
There's so many good ones. That they are considered a keystone species because they have a cascading impact on their environment. Yeah. They impact almost everything living in it. Plants, animals, bacteria, fungi, everything. The problem I have with keystone species is the majority of keystone species that we know of and that have been classified mammals. A lot of them cute mammals. Also, what is the threshold for a keystone species? Because my understanding is a lot of ecosystems are like a web, you see. And if you take one thing out, everything is impacted. So what level of impact is enough to make it a keystone species? Are we comfortable saying redwoods are keystone species? Are we comfortable saying that corals are keystone species? And then at what point aren't we just talking about species?
No,
Right.
because there are some species that definitely aren't keystone species.
Sure.
Like cats, for instance.
Cats are invasive, therefore not part of this
right.
conversation.
Right,
Well,
but
but
if
no, but I
also
mean-
if you're talking about say a lion,
Yes.
a lion is not necessarily a keystone species. It is a predator. It is the apex predator.
Or is
potentially.
it? Because
But
if you remove them,
what happens?
the hoof stock will explode in the area,
Right.
get sick and die en masse. So,
Right.
so this
Right,
is my
so
problem. Is
These are
like,
how these things work together.
this
So
is
if
my
I could-
point.
If I can make an analogy that I think is to your point also,
Yeah.
if you were to think of about something like a bicycle,
Mhm.
and what's the keystone part to a bicycle? Well, it's obviously the wheels. Oh, so you could go without the brakes? Oh no, that's a keystone part also.
Yeah, what about
so you
the
don't need a
gears?
steering wheel?
Yeah.
Oh no, you need to be able to steer so that's also a keystone part. What's the part of the bike that you could actually remove from a bike that you really don't need? The bell.
The bell.
Maybe.
Yeah.
The bell.
Oh, without you getting hit by a car.
reflecties. Oh yeah. See. Yeah. And so this is kind of what I'm saying is
Yeah.
I understand what this project is trying to do. I understand they're trying to prioritize because unfortunately we cannot save every species right this second.
Yep.
So
Yeah.
they need to prioritize.
Start somewhere.
They need to prioritize influential species that have kind of a ripple effect of an impact on a space. I get it. I'm with it. I just think we're using this keystone species definition fast and loose to fit what we want, which is like cute and fluffy usually. Also, already starting to disappear, and we're seeing what's happening as a result, right? So that's a lot of it is like the oopsie factor of, oh boy, we really shouldn't have removed that. That's important. Can you put it back? So it's important. It's important messaging. I think we should be careful about overselling the influence of some species over others.
For
Because
sure.
also
Yeah.
the lizards that live there that also dig and graze aren't considered keystone species, but they're being killed en masse by cats. So yeah, so it's very difficult to pick and choose. Anyway, back to this. I will just finish
Save
with
the
some actual
ecosystem.
science. Yeah. So what they saw as they they tried to figure out what they could do to promote rabbit populations in that area. And instead of trying to like start a breeding program or do other things that we've definitely talked about on the show in relation to bringing back endangered species, what is cool is that they found what rabbits needed to thrive in the environment and looked at why that wasn't there. So simple, cost-effective ways of encouraging rabbits includes creating piles of downed branches, they're called brush piles, and to make sure there are banks of loose soil. And so where this all comes from is actually showing that, Pip Mountjoy, Shifting Sands project manager at Natural England said, I really liked how he said it, quote, "The project's interventions have provided a lifeline for this unique landscape and shown how biodiversity can be promoted by disturbing places, not just by leaving them alone. These rare habitats are becoming overgrown and species are declining as a result of changing land management practices and human impacts. It's our responsibility to restore and maintain these spaces for nature. Some of these species exist only here, and if lost, will be lost forever." So it's a good reminder that what we think looks like a barren wasteland of a plain actually has exactly what a native rabbit might need. And when we leave those areas to go wild, we reduce our land management or we make it look more manicured. That can actually have detrimental impacts to the animals that live there. And so I I thought this was a really cool moment to recognize, like, okay, we can kind of do an audit of a space where there's an animal we're trying to make sure doesn't go extinct. And we can see, like, okay, this is the thing that they need. Okay, so we need to make sure that those things are available to them, and also that we're not doing other things here that are counterintuitive to what they need. And so I really like that look at land management, because not just this keystone species might have a positive impact from that. If that's what that plains originally looks like, there are all sorts of species that can benefit from it looking like it once did.
Yep. One species is not the only species, that they all enjoy it together. Yeah. It's a great point. Justin, do you have some stories?
Yeah. So this is actually a great one to follow. Just good news.
Woohoo!
That's that new segment where we try to show something positive from an uplifting a type of story from a topic that is usually a big downer. We try to bring you the goods version of that. This is the ecology edition. Just good news, everyone. 23 species, 23 keystone species, if you will, have been removed from the endangered species list. I just briefly looked over the list.
No.
This is actually quite impressive. It includes the little Mariana fruit bat of Guam, more than half a dozen Hawaiian island birds, one plant, 8 or 9 fish, water mussel species from freshwaters across the United States and Canada. There's the ivory-billed woodpecker, which I was amazed about because apparently this is the largest U.S. woodpecker. Hadn't been seen for like 70 years. All have been removed from the endangered species list this week. Credit for the removal has been given to developers, water pollution, logging dams, invasive pet species, feather hunters, and private trophy collectors. In each case, humans. That doesn't sound like a conservation effort, really, if you ask me. In each case, humans were the ultimate cause for the extinction of.
Yeah.
Oh, shoot! I thought this was because there says that we're getting removed from the list. I assumed.
No.
Okay, so I guess there is more than one way to get removed from the endangered species list. What's truly interesting about this is previous to these 23 getting removed, there would have only been 11 species removed from the list in this way. And some of these creatures have not been seen since long before the endangered species list was even created. And there's some sort of interesting debate about whether or not it's valuable to have the ivory-billed woodpecker that hasn't been seen in over 70 years on an endangered species list when nobody has seen it for 70 odd years. Should we still be trying to preserve a habitat? Is there financing for conservation for a thing that we've not seen? Does that money then evaporate and then development can take place because there's no more fear of disturbing the environment of a thing that's already extinct? Or is it time to face facts that these are not in danger, that we've made these 23 species extinct and that they're not coming back, and to recognize that as is an important. So some debate about that. But yeah, 23 officially, although there is a window of opportunity here.
Where they can be endangered again if they find them?
Yeah, because they've announced the 23 are being removed, but they've left like a 60-day window. If anyone can find one of these 23 species that they've been looking for sometimes 70 or more years.
In the next 2 months.
In the next 2 months, they will put them back on the endangered list.
Well, that's good news.
Yeah. So there's still some good news if you get out there and finally start looking.
Oh my.
Sorry.
Thanks for that, Justin.
Next week. I just need to read to the end next time. I just need to read to the end of the headline. I just picked the 23 species removed from the endangered species list. Sounded really
great
good.
News.
Sounded like good news.
No.
My favorite story this week Longtime listeners of the show will recall that from time to time there's a discovery which seems to fall outside of the sort of more accepted timeline for humans arriving in the Americas, which is, depending on when you learned it the first time, that date is between 8,000 and 13,500 years ago, 14,000 years ago, somewhere in that range. That later 13,500 year ago points to the Clovis culture, which has lots of arrow points that were found and then eventually dated, and some shell mounds, this sort of thing. They're in that 13,000 year range, and there's enough of those that sort of became the Clovis culture in North America, thought to have arrived after the glacial maximum had melted down, and then people had come across the Bering into the United States. This has been the sort of very popular thing. So, however, as reported on the show before, there's tool marks on bones in the cave in the Yukon. That's some 24,000 years ago. There's a cave in Mexico very recently where they found human tools, believed to be up to 30,000 years old. Multiple sites in South America that are in the 14, 15, 16, some even hinting at 40, 50, 60,000 years in age. List of outliers continues to grow, so the argument is being made, maybe they're not all outliers. Maybe we've got enough outliers at some point that the sort of accepted ranges of time need to get moved. We got to move those goalposts. And some of the things can be sort of debated. The 40, 50,000 years sites in Brazil, I think those are at the Pedra Ferrada site. They're based on charcoal that's burned and aged and human habitated sites, which could have been from natural burnings before the humans got there. There's some sites in North America that are very old too, but again, there's a carbon influence in the soil, which again sometimes can confound a bit of the dating system. Now, a new discovery has been made that should quiet any critics of the more ancient timelines. Footprints have been found at the White Sands National Park in New Mexico. Footprints made by a group of teenagers, some children, occasionally adults, footprints that are contemporary to the height of the last glacial maximum, when giant ice sheets covered much of North America some 23,000 years ago. There are tens of thousands of fossil footprints that they have been uncovering in the White Sands of New Mexico. All sorts of creatures whose tracks were deposited around the margins of large wetlands or lakes at times, and then at other times there's more of a patchwork of small water bodies that these footprints are sort of being found around. Tracks with undisturbed sediment above and below them that also contained seeds, grass seeds. These seeds then were radiocarbon dated, revealed the seeds range from the age of 21,000 to 23,000 years old, suggesting humans made repeated visits to the sites over at least 2,000 years. So this is not just some humans walking by. This is sort of persistent survival in the area. And this is concurrent with mammoths and the giant sloths are this commingling and timeline.
And they were possibly hunting them, and were they hunting the mammoths possibly? Or
so what's interesting is there's a lot of activity that looks like teenagers and children hanging out, whether at play. So it's sort of the part of the thing is that there's so much youth that are hanging out in these large groups, running around, that it has this sort of feeling of a relaxed environment. That the kids are out wandering around in the plains by the water doesn't mean that they're terrified of predators or on the verge of survival and extinction and fighting for their lives. It's more like, all right, you kids go out and play.
Let the parents take care of whatever they take care of.
Get some work done. Or, they could be out there foraging to bring stuff back to the household. but it sort of paints a much more carefree existence than one of a bunch of huddled mastodon or mammoth hunters who are, in the midst
Starving
of a
to
hunt.
death and
Yeah.
terrified and yeah.
Yeah, no, they seem like they were doing just great. Uh,
It does tell a story.
Yeah, and it feels
It
like
really
something
does.
that I can relate to. That like very
So
neat.
the amazing things about this is you have the footprints don't move. They don't. Once you get these footprints into these sands, they're not changing position. So these are some of the best examples of dating. One of the best laboratories for dating accurately, and it's putting it 21 to 23,000 years. Again, height of the glacial maximum. So this is, you got the Bluefish Cave people up in the Yukon, like waiting for the ice to thaw, trying to get down maybe. But,
Right.
but the idea had always been for the longest time that ice had to melt before humans got down. And here we're showing thousands of years of humans living below that barrier. If people are already down in New Mexico 23,000 years ago, then when could they have gotten there? How much further back in time would we need to go to actually make that possible? So, yeah, much more to learn, of course, as always.
Always more to learn. Now we just need to find, the pathway that everyone came from South America and Brazil, and then they traveled up instead of going north and down. We're just North America. Northern Hemisphere has a bias problem. We are very biased. But anyway, this is This Week in Science, and I do hope you all are enjoying the show. I have a couple more stories right here at the very end. Love bites. There are animals, and I'm not saying that love bites. I'm just saying there are animals that bite each other during their courtship. And it looks as though Tyrannosaurus Rex was one of those species.
Ouch.
Oh, wow. Yeah.
An analysis of fossilized theropod skull bones, about 528 of them in all that they looked at. 122 of them had deep bite marks and healed lesions. And they were all in skulls that were past sexual maturity. So no baby or juvenile skulls seem to have any of these facial wounds in them that they looked at. And so the researchers have concluded and published this week in Paleobiology that they think about that adults of the T-Rex species, they went into courtship very aggressively, and that the males would fight for dominance and square off against each other, biting. What they say is each animal would deliver lateral head swings to seize their opponent's skull or lower jaw. Swing that lateral head swing. So they're swinging their head to the side, mouth wide open, trying to grab on to the other dinosaur's base. And
Gently, I assume. Just very sweetly.
Oh, just a little nibble.
Very sweetly.
little love
Let
bite.
me just dent your skull a little.
Yeah. So they found that the skulls of smaller dinosaurs, other theropods that were on the branch that led to modern birds, did not have bite marks like this. And so they are making the assumption that the violence of the T-Rex theropods was left behind as birds came onto the scene and used their feathers to show off and stopped fighting each other so much.
Yeah, I don't doubt that they tried to keep biting. It's just without teeth, you can't hang on.
You can pinch,
You'd
but
be
you really
surprised,
can't grab.
is all I'm saying.
The cassowary, I think, is one of those birds that you would probably not want to let laterally swing their head at you.
You're more worried about the nail for the cassowary.
Yes,
That's the eviscerator.
That's
wow.
a dinosaur.
I feel like there's a lot of jumps happening in the story.
I absolutely agree with you.
But I'm not a paleontologist, so maybe they know something I don't. It just kind of seems like bites in the head. Yeah, that's for sex. That's it.
Yeah, it's an assumption that it wasn't in the youth, it was only in adults. And so it's being assumed that maybe courtship behaviors were aggressive. We don't know, but it seems possible. It could also be that adults just happened to get into more fights as they were scavenging for their meals.
Yeah, I mean, if you think about them like hyenas,
right?
They could fight over food all the time.
Mm-hmm.
Huh.
And they probably did.
Yeah. It just becomes more lethal with age as you become a big dinosaur.
Yeah, I thought you were going to say, like, sometimes you're fighting over food and then the mood strikes and things change a little bit.
After
the Tyrannosaurus cute, as it were.
Nah, no cuteness. Bites. Love bites. Love hurts. Yes. And then last week I talked about how, very briefly, jumped onto the topic of how dopamine is involved in making associations. So our associative memory is they've found new neurons that are involved in creating these associations in our brains. Dopamine is tied into that. This week, research out of UC Davis has looked at how the brain's memory center, the hippocampus, activates during the learning of stories and then the recall of stories. And what they discovered is that, yes, indeed, the hippocampus is your brain's storyteller. It pieces together bits of information, events that are taking place in space and time with all their contexts, and links information to help you put together an entire story. The way that they determined this was looking at people under functional magnetic resonance imaging. They actually were watching the activation of the brain as they were telling people stories. And they would tell people related snippets of stories and then unrelated snippets of stories. And the people would put them together and figure things out and then have to remember them. But the way that it all worked is they would start with something, say like, Beatrice calls to ask for advice about a dilemma with her partner. Melvin gets locked out of his home in the middle of cooking a pizza. So these are two completely unrelated
So Melvin's
events.
the partner who's locked out. That's Beatrice is like, hey, how do you get into a house? My partner can't get.
Right. So if you in your
Start
brain
building it.
put together those two pieces of information as a whole story, your hippocampus is right now, as you're putting it together, activating to link bits of information. And so in their study, hopefully people weren't putting Beatrice and Melvin together, but what they were linking later was another bit of information about Beatrice, that Beatrice shows up wearing a cast on her arm after falling off her bicycle. So you know that there's Beatrice is having an issue. She calls about a dilemma, and then she, maybe has said she's going to come over and talk to you, but something happened during the day, and oh my gosh, you got hurt on your way over to my house to talk about this problem with your issue with your partner. You put together all these contextual bits of information, and it's the hippocampus that becomes activated to make that happen. It does not activate as much for completely unrelated bits of information. So, and in recall, if you are recalling information, once again, the hippocampus gets activated to bring all these bits of information that it has helped network together. It's like the connector for the information that has been stored in a distributed manner in your brain. So those association neurons that were stimulated by the dopamine that we talked about last week, that have associated a smell with a certain memory or a fact, or, a piece of information with something else, they're getting triggered. Those little neurons, the association neurons, are getting told by the hippocampus that they're now going to be talking with these other neurons that have another bit of association or another piece of information from a different part of the brain. So the hippocampus is really a connector and a storyteller of narratives. Without a hippocampus, you can't hold a story together. And as I was thinking about this story, it made me wonder if the aha moment where you suddenly put disparate pieces of information together in a new way, if that is in some way related to an activation of your hippocampus, allowing a new networking, a new association of different bits of information.
That's cool. It's like the Sherlock Holmes, connection. Yeah. Is the hippocampus, that's what I want. I'd love to know that. And we don't have any way necessarily of knowing that at this point in time, unless we get people to have aha moments while we've got them in an fMRI machine. But
I think that's possible. Yeah.
Have them watch a mystery movie.
Yes, a whodunit.
Yeah.
And when you figured it out.
Yeah.
I know who did it. Yeah. Our brain, it's so fascinating to me that we have these pieces of neural tissue that once upon a time, to go back to the book that Bill Schutt has written about the heart we were talking about, once upon a time, the brain was just tossed away. It was considered a bunch of goo. And now we're understanding that it is this very specialized organ with these specialized nuclei that process information and allow us to analyze information, to remember things, to create stories, and to live understanding metaphor, and to live as storytellers of our own lives. Our brain helps us do that. And it's just, thanks, brain. You're awesome.
Yeah. Couldn't do it without you, buddy.
Couldn't do it without ya. And on that note, I can't do it without you. This show. Thanks, Blair. Thanks, Justin.
Thank you.
I think we've made it to the end, yeah?
Yeah.
Another episode. Wipe my hands. Get the chalk dust off of them. No, I don't have any chalk or anything. I've got lots of computer files. And I want to say thanks to all of you out there for joining us for another episode. Thank you for enjoying the science and for having the questions and for being curious and coming back week after week. Thank you to Bill Schutt for joining us to talk about his book. I put it down, Pump, The Natural History of the Heart, which is available in bookstores now. Thank you to Fada for help with social media. Thank you to Gord and Arnlor and other people who are involved in moderating our chat rooms for helping to make sure our chat rooms are safe and pleasant places to be. Thank you to Identity4 for recording the show. And thank you to Rachel for helping with editing and also for your assistance. It's really wonderful. Thank you to our Patreon sponsors for all of their support of the show. Thank you to Kent Northcote, Rick Loveman, Pierre Velazar, Ralph E. Figueroa, John Ratnaswamy, Carl Kornfeld, Karen Tazi, Woody M.S., Andre Bassett, Chris Wozniak, Dave Bunn, Vegard Shefstad, Hal Schneider, Jonathan Stiles, a.k.a. Don Stylo, John Lee, Ali Coffin, Maddie Perrin, Gaurav Sharma, Shubru, Don Mundus, Stephen Alberon, Daryl Myshek, to Stu Pollock, Andrew Swanson, Fred S104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles Jack, Brian Carrington, Matt Bass, John and Nina Lamb, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard, Brendan Minish, Johnny Ridley, Kevin Railsback, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Hey Arizona, Support Aaron Lieberman for Governor, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Mallory Sutter, Philip Shane, Kurt Larson, Mountain Sloth, Jim Drapeau, Sarah Chavez, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Disney, David Simmerly, Patrick Pecoraro, Tony Steele, Ulysses Adkins, Brian Condren, and Jason Roberts. Thank you for all of your support on Patreon. And if you would like to support us on Patreon, you can head over to twist.org and click on that Patreon link on next week's show.
We will be back Wednesday, 8 p.m. Pacific, broadcasting live from our YouTube and Facebook channels and from twist.org live.
Do you want to listen to us as a podcast? Maybe while you bake a souffle? Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you've heard here today, show notes and links to stories will be available on our website, which is www.twist.org, and you can also sign up for a newsletter.
You can also contact us directly. Email Kirsten at kirsten@thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put TWIS, TWIS, in the subject line, or your email will be spam filtered into the mouth of a T-Rex who's feeling romantic.
Oh, nice. If you want to avoid romantic T-Rexes, you can just hit us up on the Twitter, where we are at Twist Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you'd like us to cover or address, a suggestion for an interview, a haiku that comes to in the night, please let us know.
We'll be back here again next week, and we hope you'll join us for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world, so I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice, show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand, and all it'll cost you is a couple of grand. 'Cause this week's science is coming your way, so everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way, so everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 science.